students, welcome to my virtual classroom, and for today's discussion in biochemistry, we will talk about proteins. Okay? What comes in your mind when you hear the word proteins? So in biochemistry, we define protein as an organic polymer composed of amino acids bonded together in one or more chains. And there are different structures of proteins. And proteins play a vital role in our body. An amino acid has a central carbon atom to which are bonded to a carboxyl group. An amino group, a hydrogen atom, and a variable side chain designated as R. Okay. Just like this structure that I'm going to show to you. So this is a simple structure of an amino acid. Okay. So we have the amino group and the carboxyl group. If you can still remember in your organic chemistry, when the functional group is a carboxyl group, so alam natin na acid siya. Okay? But there are also some structure na even meron siyang carboxyl group. Depende pa rin yan kung ano yung mass priority lalo na kapag when it comes to naming. So we have to follow the EUPAC or the International Union of Pure Applied Chemistry. Okay, mayroon silang names. Okay, the structure of amino acid. So an amino acids bond to each other by forming a peptide bond. An amide group formed by a condensation reaction between the carboxyl group of one amino acid and the amino group of another. Okay. So we have two, as we have here in our presentation, we have here a structure of a dipeptide. Okay. Two amino acids linked by a peptide bond form a dipeptide. So a chain of two or more amino acids peptide bond is called a peptide. Okay? The term polypeptide is applied to a chain of 10 or more amino acids. Okay? So proteins may have one or several polypeptide chains, and each chain must have may have one or several polypeptide chains. And each chain must have an exact sequence of amino acids. Okay. So proteins can be fall into either round globular structures or long, such as the fibrous structures. And we have two different simple kinds of protein. We have the globular protein and the fibrous protein. The amino acid chains are held in place in three-dimensional structures by attractive forces between the side chains of different amino acids, which have been brought close together by the bending and folding of what we call this of the polypeptide chains. Okay, we have here now enzymes. So many of the proteins in an organism act as a enzyme or act as an enzyme. So these proteins catalyze chemical reactions, speeding up reactions or allowing the reactions to take place at a low temperature. So enzymes action. So substrates are brought close together in active sites of an enzyme, which lowers the activation energy of the reaction by facilitating the bonding together of the substrates to form a product. Okay, based on the illustrations that I have here, we have here a substrate. Okay, it will bond now to the active site of the enzyme. Okay, and eventually they will form a product. Now let's talk about carbohydrates. Carbohydrates include sugars, starch, and cellulose. So carbohydrates contain carbon, hydrogen, and oxygen in a 1:2:1 ratio. Now let's talk about monosaccharides. So the simplest carbohydrates are just simple sugars, or also known as monosaccharides, which commonly have 5 or 6 carbon atoms. So glucose, the main ingredient in corn syrup, is a family. It is a common monosaccharide, common sugar. So monosaccharides, glucose has the molecular formula of C6H12O6 and can be presented by the following structures. Okay? We have here in a cyclic form, and we have also the open chain form. This is the structure of glucose. Monosaccharides. So the most common simple sugars are glucose, fructose, and ribose. Okay? Kanina, we have the structure of the glucose, and now we have the structure or the illustration of fructose and ribose. Polysaccharides. So a polysaccharides, it is a polymer of many monosaccharides bonded into a chain, is called a polysaccharide. So starch is a polysaccharide that consists only of glucose units. Okay. So plants also link glucose units together in a different way to form the polysaccharide cellulose, which forms plant cell walls. So animals store glucose as a polysaccharide called glycogen, which is similar to starch. Okay, we have here a glycogen. Now let's talk about lipids. So lipids are the nonpolar substances which is also known as, for example, we have the fats, the waxes, and oils produced by living things. So lipids are not polymers, and their chemical structures vary widely. So lipids contain carbon and hydrogen in a 1:2 ratio with only a small amount of oxygen. Next, we have the fatty acids. The most familiar lipids are the plant oils and animal fats. So these lipids are esters of fatty acids, which are carboxylic acid with long, straight, and hydrocarbon chains, usually having between 12 and 24 carbon atoms. So we have here now the illustration of a saturated fatty acid. This is a stearic acid. So the simplest fatty acids are the saturated fatty acids, which have no double bonds between carbon atoms. So stearic acid is found in pork and beef tissue. We have here a monounsaturated fatty acid. So many other fatty acids have one or more double bonds between carbon atoms, and as a result, they become unsaturated fatty acid. Oleic acid is a major component of olive oil. Okay, sometimes this is read as oleic acid. Triglycerides. Animal fats and plant oils are made up of primarily of triglycerides. Okay, that is triglycerides. Molecules in which 3 fatty acids are bonded to a glycerol molecule by ester linkages. Okay, so we have here a structure of a triglyceride. Then we have also other lipids. Okay, another class of lipids, we have these steroids, consists of compounds whose basic structure is very different from those of other liquids. Okay, we have the cholesterol, vitamin D, and some hormones are steroids. So this is the basic structure of a steroid. Okay, what is the function of lipids? Lipids have 2 major biological roles in the body. First it is an excess energy absorbed by an organism can be stored as lipids. This is why humans store body fat if they eat too much or move too little. And the second one, it is used as a chemical messenger. Okay, and these chemical messenger are called hormones. So many steroids are hormones. Next, we have the nucleic acid. So the sequence of amino acids in a protein is determined by the genetic information coded into long-chain polymers called nucleic acid. Okay? The monomers that make up nucleic acids are called nucleotides. Again, the monomers that make up nucleic acids are called nucleotides. So each nucleotide is made up of 3 parts. First, the phosphate group. The second one is a 5-carbon sugar. And last is a nitrogen-containing cyclic compound called a nitrogen base. Okay, so we have here a structure of a nucleotide. Okay, so we have the phosphate group, the nucleotide, and the nitrogen-containing base. Okay, nucleic acid. So ito yun. Phosphate, sugar, then the base. Okay. We have 4 nitrogen bases. So we have the adenine, cytosine, guanine, and thymine. Okay. Next. So the common nucleic acids are DNA. So again, the common nucleic acids are DNA, or that is deoxyribonucleic acid, and RNA, or the ribonucleic acid. So DNA contains the sugar deoxyribose, and RNA contains the sugar ribose. So in DNA, the base thymine bonds with adenine, and while in RNA, thymine is replaced with uracil. So DNA is a double strand, while RNA is a single strand. Okay, we have here a structure of DNA. So this model of a portion of a DNA molecule clearly shows its complexity. So a single DNA molecule contains many thousands of nucleotides. Base pairing. Okay, so the two sides of DNA are held together because the nitrogen bases of one chain are hydrogen bonded to the nitrogen bases of the other chain. So we have 4, as what I have said earlier, we have 4 different nitrogen bases are found in DNA. We have the adenine, guanine, cytosine, and thymine. So adenine nitrogen bonds thymine, while the guanine hydrogen bonds to cytosine. So the order of these 4 nitrogen bases along one of the DNA chains provides the information for the sequence of amino acids in proteins. So cell mechanisms read the DNA sequence in groups of 3 bases called triplets. So each triplet codes for a specific amino acid or tells the cell to start or stop making a protein. Let's talk about types of proteins. So we have 2 kinds of proteins. We have the globular proteins and the fibrous proteins. So enzymes are globular protein, and the shape of the globular proteins, compact shape like a ball with irregular surfaces, while the fibrous proteins usually span a long distance in the cell. So it has a 3D structure, is usually long and rod shaped. The importance of fibrous protein for the intermediate filaments of the cytoskeleton. So the structure scaffold inside the cell. Okay, the keratin in hair, horns, and nails. So those are examples of fibrous proteins. And for the extracellular matrix, so bind cells together to make tissues, secreted from cells and assembled in a long fibers. So we have the following fibers here. So we have the collagen. It is a fiber with a glycine every 3rd amino acid in the protein. And the second one is the elastin. unstructured fibers that gives tissue an elastic characteristic. Proteins at work. So the conformation of proteins gives it a unique function. So to work, protein must interact with other molecules, usually one of a few molecules from the thousands to a one protein. So ligand, the molecule that a protein can bind, and the binding site, it is the part of the protein that interacts with the ligand, and this consists of a cavity formed by specific arrangement of amino acids. Okay, so we have here is the illustration of the ligand binding. Okay, the ligand, and we have here this non-covalent bonds and the binding site. Okay, so it fits. So the ligand in the binding site, and we have here these non-covalent bonds. The antibody family. So a family of proteins that can be created to bind to almost any molecules. Antibodies, also known as immunoglobulins, are made in response to foreign molecules. So example, we have the bacteria, the virus, and even pollen. These are called the antigen. So bind together tightly and therefore inactivates the antigen or marks it for destruction. So the antibodies, so Y-shaped molecules with two binding sites at the upper ends of the Y. So the loops of polypeptides on the end of the binding site are what imparts the recognition of the antigen. So changes in the sequence of the loops make the antibody recognize different antigens. Next, let's talk about lysozyme. Okay, lysozyme. So, lysozyme is an important enzyme that protects us from bacteria by making holes in the bacterial cell wall and causing it to break. So, lysozyme adds water, H2O, to the glycosidic bond in the cell wall. So, a lysozyme holds the polysaccharide in a position that allows the water to break the bond. This is the transition state, which state between the substrate and the product. So active site, it is a special binding site in enzymes where the chemical reaction takes place. We have here now the structure of the lysozyme. Non-covalent bonds hold the polysaccharide in the active site until the reaction occurs. So the features of this enzyme catalysis. So enzyme binds to two substrate molecules and orients them precisely to encourage a reaction to occur between them. Next, the binding site of substrate to enzyme rearranges electrons in the substrate, creating partial negative and positive charges that favor a reaction. And lastly, enzyme strains the bound substrate molecule, forcing it toward a transition state to favor a reaction.